Welcome to our video infographic on proteins, essential biomolecules for life. Let's explore their detailed structure, functions, and the fascinating world of amino acids. Proteins are made up of long chains of amino acids, the building blocks of life. There are 20 different amino acids that combine in various ways to create proteins. Amino acids have a central carbon atom, an amino group, a carboxyl group, and a side chain. The side chain or R group determines the identity and properties of each amino acid. Proteins perform numerous roles: enzymes, hormones, antibodies, and structural components. The unique sequence of amino acids defines how proteins fold and function in the body. Protein structure is typically categorized into 4 levels: primary, secondary, tertiary, and quaternary. The primary structure is simply the sequence of amino acids in a polypeptide chain. Next, we have the secondary structure that forms through hydrogen bonding between backbone atoms. Common secondary structures include alpha helices and beta sheets, providing stability. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain. Tertiary structure involves various interactions like hydrophobic interactions and disulfide bonds. Finally, quaternary structure describes how multiple polypeptide chains come together. Hemoglobin is a classic example of quaternary structure with its 4 subunits working together. Now let's discuss the different kinds of amino acids based on their side chains. Amino acids can be categorized as non-polar, polar, acidic, or basic based on their properties. Non-polar amino acids are hydrophobic and tend to be found in the interior of proteins. Polar amino acids are hydrophilic and often reside on the surface of proteins in aqueous environments. Acidic amino acids have side chains that donate protons and carry a negative charge at physiological pH. Basic amino acids possess side chains that can accept protons and carry a positive charge at pH 7.4. Each type of amino acid plays a vital role in maintaining protein structure and function. Additionally, modifications such as phosphorylation affect protein activity and regulation. Enzymes are proteins that catalyze biochemical reactions essential for metabolism. Their activity relies on their unique structure, which allows them to bind specific substrates. Antibodies are another crucial type of protein that helps defend against pathogens in the body. Lock and key model describes how enzymes specifically fit into their substrates like keys in locks. Induced fit model suggests enzymes undergo conformational changes upon substrate binding. Denaturation refers to the process where proteins lose their shape due to extreme conditions, e.g., high heat or extreme pH. This impacts protein functionality significantly. Chaperone proteins assist in proper folding to ensure correct protein structures are achieved. Misfolded proteins can lead to diseases such as Alzheimer's or cystic fibrosis in some cases. Protein synthesis occurs via transcription and translation processes within cells and tissues. Transcription creates mRNA from DNA that carries the code for protein assembly. Translation happens in ribosomes where mRNA is read to assemble amino acids into proteins. Post-translational modifications can further influence protein functionality after translation occurs. Glycosylation is one such modification where carbohydrates are added to proteins for function. Proteomics is the study of all proteins expressed by a genome, providing insights into biology. Structural biology examines protein structures to understand their roles and mechanisms. In summary, understanding proteins reveals crucial aspects about life processes and diseases. Thank you for joining us on this journey through protein biochemistry today. For more educational content, stay tuned for future videos.